Okay, hello everyone. My name is Samir, and today I'm going to be talking about plasmonic metasurfaces for spectrochemical tissue imaging. So my research operates at the intersection between nanophotonics, mid-infrared imaging, and molecule detection by absorption spectroscopy. So more specifically, what we are doing is we're benefiting from extraordinary light-matter interaction at the nanoscale to do biomolecule detection. So since I want you to understand how to use plasmonic metasurfaces for spectrochemical tissue imaging, I want you to imagine you have an optical microscope, and as in any optical microscope, you need a light source. So it happens that in our situation, the light source is a mid-infrared laser. And then what happens when you shine light with a mid-infrared laser into a biological sample is that you can capture the absorption. And that's what I'm presenting here. That's an absorption signature. Now, all these fluctuations that you see in the absorption signature, they are going to tell you something about which molecules are present in your biological sample. So when you image the biological sample, they are going to start, the molecules between the biological sample, they are going to absorb light, and they are going to do one of these 6 things that we are seeing here. And then depending where the oscillation or the fluctuation is happening in the absorbance signal, you can classify that molecule as a lipid, a nucleic acid, or a carbohydrate. More formally, you can describe the absorption of a biological tissue sample using Beer-Lambert's law. So what this law tells us is that you can find the absorption just by calculating the logarithm between your incoming illumination and your outgoing illumination. What this actually means is that if you know your absorbance, you can also have access to the concentration of the particular molecule that you have there, and then that you're looking information, right? As with any imaging technique, there are some limitations that are inherent to the physical properties of your sample, and some limitations that come from your optical system. So how much light is going to pass through, and how much light is going to make it in your detector. So the fundamental limitation of conventional mid-infrared spectroscopy is that it yields intermingled information between the morphology of the sample and the chemistry of the sample. So just as an example, we have imaged 3 different thicknesses, 20, 40, and 80 micrometers tissue samples in transmission mode. And then what we saw is that if we try to quantify the composition of the molecules that we are targeting in the tissue sample, there is a linear trend. So with this, we don't know if the concentration is coming because the absorptivity is more, because we have more concentration of the specific analyte, or because we are just doubling the thickness of the sample. So there is some ambiguity there. What we propose is to use plasmonic metasurfaces. So we call this method metasurface enhanced mid-infrared spectral tissue imaging, CCBIR. And then what I'm showing here is simplified version of our optical setup. So we have a quantum cascade laser that is going to send light towards the sample, and then we are going to read from the back of the metasurface. And then we are going to collect all that information as a hypercube. Now, because we are capturing information from the interface between the metasurface and the tissue, we are going to be able to see those fluctuations. And as I mentioned earlier, if we know the fluctuations, you have access to the concentration. So this is how the metasurface looks like, and then this is how metasurface plus brain tissue looks like. So when we measure the spectrum from the metasurface, we are going to see the response that is here in the dashed line. And then once we put the brain tissue on top, and we just take 1 pixel of the hypercube, we are going to see those dips. And then we can correlate those dips to vibrational in the molecules that the tissue are made of, right? So because we are exclusively capturing light-matter interaction at the nanoscale, now we don't see linear trends anymore, regardless of the thickness of the tissue sample. So we see the concentration for 20 and 80 micrometers tissue samples. They are along the same values. Why? Again, because we are capturing just only the interaction between light and matter at the nanoscale. So if we want to do that, if we want to do this using metasurfaces, we have to design the metasurface. So we have designed this metasurface, and that metasurface has around 3 million meta units, and we call this a meta unit, and that's our unit cell. So this unit cell has the shape of a letter U, L-U, we call it ULU. And then what happens with this specific structure is that when you shine mid-infrared light, it is going to light up at some specific wavelengths or wave numbers. And then it has two polarizations. And having two polarizations or two modes is important because in the 90 polarization, we are going to see two peaks. And then in the zero polarization, we are going to see one peak. So pretty much we are enhancing the spectral response of the metasurface across the range of interest. So what this technique has to offer is that we can resolve molecular vibrations in a better way. So the first peaks that are happening in the 90 polarization, they are going to resolve this band and the protein band. And then the second peak, which is happening in the zero polarization, is going to resolve this region of the spectra. So what CCBIR is capable of doing is that we can resolve more molecular vibrations in a better way. And those molecular vibrations are coming from the nanoscale volume. Finally, another thing that the metasurface has to offer is that it can capture small amount of tissue samples. By small here, I mean a couple of nanometers. It ranges from, I don't know, 10 nanometers to 100 nanometers. So when we just go with a standard substrate and we try to see what kind of signal is coming from these dark regions, we see that pretty much the signal is flat. But when we go and analyze the same region of brain tissue that is on the metasurface, we see these dips, and these dips are being enhanced because of the action of the metasurface. So we believe CCBIR can help identify protein conformational changes. Why? Because we are decoupling the morphology of the tissue from the chemical information. And in cancer research, it can be useful to elucidate the TME changes such as fibrosis and collagen morphology. I'm going to switch gears here. This is a second project that we are doing in our lab. So instead of using conventional mid-infrared spectroscopy instead of using CMIRC, we are going to use just MIRC, which is conventional spectroscopy. And then what we're trying to do here is to image collagen on pancreatic tissue samples. And then the idea here is that we want to use MIRC because we can do this in a faster way. Faster way, I mean 1 minute. We can scan 650 by 650 field of view. So the approach that we are following is that we take the pancreatic tissue samples, we mount them in a calcium fluoride substrate. Why calcium fluoride? Because calcium fluoride is transparent for infrared light. And then we image the samples using MIRC, and we image them using a second harmonic generation microscope. And then basically we can combine these two approaches. We can train our model, and then we can predict how the distribution of collagen is. And then we can compare that to the ground truth, which is going to be the signal coming from SHG. So these are some preliminary results in which you can see that just by using MIRC, we can see the distribution of collagen. And kind of like you can see here that this distribution of collagen looks a lot like this one. With this, I conclude my talk. I have to acknowledge my PI and all our collaborators, Dr. Liserich, Dr. Chao, and Dr. Michael Katz, same as my lab mates, the funding resources, and all facilities that help us developing this metasurface. Thank you.
Maybe I missed it. What type of enhancement do you get with the metasurface?
So the main idea is that when you try to image tissue sample using conventional mid-infrared light, you are going to go through the sample, right? And then you are going to have absorptivity, you are going to have the concentration, and you are going to have a change in the path length, right? So all these three components, they come in your signal, and then you know which one is affecting your measurements. So the enhancement that is happening with SIMMERCY is that pretty much you shine light into the metasurface, and then that light is going to couple to this structure. And then because of the coupling between mid-infrared light and the structure, it's going to create basically an electric field around the structure, right? And we know the shape. This is, I'm being a little bit not rigorous here, but you can think of the shape of that electric field like this, right? Now, if you have a sample on top of that electric field, and that electric field is being, let's say, changed because now you put a brain tissue sample there, now you are going to capture that information from the nanoscale volume, because the electric field is not going everywhere in the vicinity of the metasurface. It's just going to go 100 nanometers on top of the metasurface. So with this, basically, you are just neglecting the morphology of your tissue sample. You are just capturing the information that is within those 100 nanometers. And that's where the enhancement is happening.
Yeah, I guess, what's the fold enhancement within that volume?
So pretty much, if you have no metasurface, you send light, you get light, it's going to be 1 or less than 1. Best case, you will get 1. Why? Because whatever you're sending, that's what you're collecting. So 1 divided by 1 is 1. Now, if you don't have, that's the case when you don't have metasurface, you get 1 or less than 1. When you have metasurface, the electric field enhancement is going to be, it's going to change with e to the power of 2. So that's the enhancement we are offering with this technique. Mm-hmm. That does answer your question.
Yeah, that was really
Okay. Okay.
Just real quick, I saw you could go brain sections up to 80 microns. How thick can you make tumor sections?
So pretty much this technique just doesn't care about the thickness. And that's the beauty of the technique. You can put 20, you can put 40, you can put 80. Doesn't matter. Why? Because we are capturing just information 100 nanometers above of the surface of the metasurface. So yeah, you can do any tissue sample. Thank you.
Next up will be the ACS patient advocate.